thank you for inviting me to speak here today. It's great to be representing the Huttenhauer Lab. I'd like to talk to you a little bit briefly about two projects that are currently going on in the lab. One is directly related to interaction between the human microbiome and our surrounding environment. And then the other is more of a methodological piece that we're working on now, which I think is relevant to microbial studies in general. So I'll touch on both of these today. So this is a map of Boston's public transportation system. And it has a daily ridership of about 1.3 million people. I am among them. And this represents to us a great model system for investigating the relationship between the human microbiome and that of the urban environment, where you have 1 million people that are all funneling into this small system and touching all the surfaces there. And so we have, with support from SLOAN and cooperation of the MBTA, started a project to try to understand better the relationship between the human microbiome and that of the MBTA. So the goals of this project, there are a few of them. The first one is just to try to find out what exactly is living on the T. This is something I've been thinking about for as long as I've been actually riding on the T, and now we'll actually try to find out what that is. So more specifically, what we want to know is what are the bugs that are actually coming off of the human skin surface, getting onto surfaces in the transportation system, and then staying there and becoming stable communities. And there are 3 surfaces that we're primarily focusing on right now. Those are in the picture here. The handholds, the things that you'll be holding on to make sure that you're not falling over while you're on the train. Or if you're lucky enough to have a seat, then the seat's there as well. So both of those first 2 surfaces are actually on the train cars that are moving around through different communities, interacting with lots of different people across the metro area. And then also these terminals, like the one that you're seeing at the end there, which is what you use to buy your ticket or to add value to it. And these are touchscreen interfaces, so you have a lot of people contacting those as well at different points around the city. So that's the first idea, is to try to figure out what are we actually putting onto these services. And then the second goal of this project is to find out whether it's possible then for these stable communities that are established on the surfaces to be transferred back onto a person that's now touching them and alter that person's microbiome. So in other words, am I actually picking things up off of the T, which then change the flora that are on my own hand, for example. And lastly, sort of related to both of these goals, is what understanding if these 2 sorts of transfer processes are happening, what is going on to actually modulate them, or what can we do to modulate them in terms of how trafficked the surfaces are, how often they're cleaned, and also choice of materials in terms of if we're thinking about what we want to recover these seats with the next time there's some sort of a renovation, what kind of influence that has on movement of the microbiome back and forth. So this project just sort of started earlier this year, and we're currently in a massive evaluation phase where we're looking at different techniques to actually carry this forward. We had a great team of rotation students that ran around on the T in, I think it was January, and there are some more out now today as I'm speaking, evaluating things like surface swabbing materials and the techniques, everything from pressure to repeated swabbing, and then seeing later how those affect our ability to extract and amplify DNA to do sequencing on these. And there's also been a lot of evaluation just as sort of logistical matters. If you want to get all of the sampling done across the MBTA system as quickly as possible, but it's actually spread out quite a bit in time and space. This is actually a nice map of the MBTA spread out in time to show you how long it actually takes to get between different points. So it's been quite an adventure doing that, and the MBTA has been really helpful for us sending along MBTA police officers with our people who are collecting specimens, because we find that one of the things that has also been something we had to evaluate and troubleshoot is how the public on the trains is interacting, responding to the fact that there are technicians who are swabbing them and putting things into coolers and so forth. So all of that's in development right now, and this is mostly just a preview of this project. I'm sure next time this meeting comes around, we'll have a lot more to talk about. But I'm really interested to hear feedback from all of you and communicate with you more about ideas for this project in the second and the latter parts of the conference. So the second thing that I wanted to talk on has really been my focus since I started in the Huttenhauer Lab last October, and that's on the relatively new field of metatranscriptomics, so looking at the RNA composition of bacterial communities. And in this case, we're looking at samples that are derived from humans, but the concepts I'll talk about and the ideas are easily applicable to other microbial communities. So this is just sort of general biology, which we happen to be talking about in the context of the human body, but it's really bug biology and not human biology. And so I'll talk to you a little bit about this. So this work stems from a pilot project we're working on with the HPFS cohort, which is a group of 50,000 health professionals who have been followed since the mid-'80s for all kinds of different health-related biostatistics and epidemiology projects, and we're now getting into doing some microbial analysis on these groups. So the data that I'll talk about today are from 8 volunteers, or eight members of this cohort, where we've taken saliva and stool samples and then done metagenomic sampling, and sequencing rather, on the bacterial communities from their saliva and stool, and then also metatranscriptomic sequencing from their stool. So we were able to assay both the DNA in the saliva and the stool, and then also the RNA that's being produced by the stool community. For non-biologists in the audience, just to explain a little bit about what RNA is, if you think about DNA as being a blueprint for an organism or a protein or something like that, something that an organism is trying to build, the analogy that I like to think about is the RNA is like a Xerox of that blueprint that you're sending out to the construction site, telling them this is what you're going to work on today. So whereas the DNA is giving you something of an idea of the potential of what's going on in this community at a given time, or for its history, the RNA is really telling you what are you doing right now. And so we're interested to know what RNA can tell us. So if you profile the DNA, the genomic content of a microbial community, in this manner, what you can do is figure out something about its functional potential. So here in this cartoon diagram that I've drawn, we have 3 genes that are showing up in this community, A, B, and C, and we see that there are more copies of gene C showing up than gene A. So we would say that there's more potential activity for gene C. Again, this is all about potential. When we look at the RNA data, there's a few different things we can see. Sort of the simplest possibility is that every bacterial gene is just cranking out a certain amount of RNA all the time. And so in this case, activity and potential are the same thing. But there's other possibilities. One of them is that some of these genes aren't actually active at a given time. Now this is sort of standard biology for multicellular organisms like human, where my brain cells and my gut cells have the exact same blueprint, but they're doing different things at different times. But we don't actually know for these the gut microbiome if the same sort of thing is true. So we can imagine that there are cases of genes that we see them at the DNA level, but they're just not really be showing up at the RNA level. One case of potential not being the same as activity. Conversely, we can see genes that are very sort of minor components of the community at the DNA level, but they're very highly expressed. So they form a larger percentage of the total RNA of the community. And so these are all possibilities, and we wanted to investigate what we actually see. So these are the real data for those 8 individuals for the gut, where we're actually, each of these dots is a gene, and we're correlating the amount of DNA we see for that gene with the amount of RNA that we see. So the DNA is on the horizontal axis and the RNA is on the vertical axis. And so we can see a few things from this plot, one of which is that for about 50% of the genes, there's a pretty strong correlation, meaning that a lot of genes just are on all the time, cranking out about as much RNA as you would expect based on their DNA level, with some fluctuation above and below that line. So there are some genes that are making a little more RNA than we would expect, and some that are making a little less. And we also see these clusters of genes on the side, where this is a case of genes where we only see RNA. This gene was never detected across the 8 individuals in their DNA, but we do see it at the RNA level. And on the other axis, we have genes that we only see their DNA, and we don't see their RNA. They don't appear to be expressed. So we've dug into these a little bit to find out what's going on, and the stories make a lot of sense. So among the genes that are sort of highlighted in blue, where they seem to be less active than we expect from the DNA, we see things like amino acid biosynthesis, pathways for making things. In this case, potentially things that bugs don't need to be making. If the amino acids are very rich in the gut environment, then it makes sense that these pathways wouldn't be on in a lot of the bugs. And also things like certain stress response pathways, where if the bugs are actually happy in this native environment, then we wouldn't expect them to actively be mounting a stress response. And that's sort of traditional biology of things that just aren't being used, so they're being shut down. On the other side, we have these genes that are much more RNA active than we would have expected. And from what we can tell so far, all of these have to do with archaea that are living in the gut. Methanobrevibacter smithii is one of these that shows up in a subset of the population, and it metabolizes methane, it has its own ribosome, and these things seem to be showing up a lot in the RNA. So basically, these bugs are there, but they're much more active than we would expect from their relatively low DNA concentration. So this is something that we're learning about genes from looking at RNA. We can also learn some things about bugs, as in the case of the Methanobacter species. We can see something about the way the bugs are behaving in the system. We can also learn some interesting things about the way that bugs move around in the human body. So here we're looking at a heat map of bugs that are found in the oral and gut communities. And one thing that's of great interest to the dental researchers that we collaborate with is this idea of transit, these highly abundant oral bugs that tend to show up in the gut community. So they're surviving the trip down there, and then we're actually able to detect them in the gut. However, when we actually look at this picture from the RNA perspective, although we can see these bugs that are supposed to be in the oral community showing up pretty strongly at the DNA level in the gut, most of the time they're not very happy about being there. And we can see that from the RNA, that a lot of them, they're just not really metabolically active. With one exception, this Streptococcus salivarius, which comes from the oral community and then seems to do quite well and be quite active in the gut. So this is something that we can learn about bugs and whether bugs are actually active and happy about their environment from RNA data. And lastly, is a story that RNA can tell us about people, or more generally about the environments that these bugs live in, which for our case happens to be within people. One of the things that I find most fascinating about a lot of the results coming out of microbiome research is that while a body site might vary from person to person a lot in its taxonomic composition, so here we're showing the relative composition of the genera across the 8 individuals in this cohort, their functional potential is very flat. We see that they tend to be functionally very similar in terms of the things that these bugs are capable of doing, even though their names are actually different from one bug to the next. And so we can actually quantify that with a measure like Bray-Curtis dissimilarity, and you see that when you compare people, they tend to be quite different at the genus level, but they are very, very similar at the level of these enzyme categories. When we add the RNA data in, we see that there's a little bit more differentiation from one person to the next when we look at the actual functional activity that's going on. So it's not nearly as diverse still as the taxonomic composition, but it's more diverse in potential. So what we're actually seeing is that although these bugs are all capable of doing the same thing, it seems, they're doing different things to different degree. And this makes a lot of biological sense, that even though these bugs may have the same sort of cassette of genes they're working with in one individual or another, because each individual represents a specific environment, they can tune the activity of those functions by changing RNA levels to better adapt to where they find themselves. And so that's something that RNA is actually able to tell us about bug environments and communities. So just to sort of summarize these two projects quickly, we have this one project where we're looking at RNA data. And again, we're looking at people, but I think that this is a very sort of extensible idea, that if you want to know more about activity and less about potential about bugs in an environment, whether it's the human body or a building surface, that RNA can tell you a lot about that. And then we also have the project going on that I previewed at the beginning, where we're looking at the biology of the interaction between host and or human and urban environment. The T is the example. And I hope to communicate more with all of you about that today and come back to the lab with some ideas for continuing that project. So thank you for your attention and invitation.
So that's a really interesting result about the differences in the transcriptome composition versus the metagenome composition. Are the bioinformatics processing pipelines exactly the same for those two?
So far, yes, we're treating them entirely the same. So I come from the bioinformatics background. I'm actually developing a lot of the work or the tools we're using to analyze this downstream. Mm-hmm. And we get raw sequence data back for both of them, and then
and you annotate against the same databases.
That's right, exactly. Yeah. So in the case of looking at the genes, we're using a tool called Human that was developed in the lab to quantify the levels of those genes at both the DNA and the RNA level.
Mm-hmm. Cool.
I also thought that metatranscriptomics was really interesting next to the metagenomics. And it's hard to do in indoor microbiome, but do you have any plans for something like that in your subway samples?
I think for right now, the subway samples are going down a 16S pipeline. So in that case, metatranscriptomics wouldn't be happening. I don't know. It's a really good question. I know even in the saliva community, there have been technical hurdles that have prohibited looking at some of the RNA data there. It was a lot easier to extract it and work with it in the stool. All of that happens upstream before I pick up with the data, so I'm not entirely sure what that is, but I know that it's not easy, so it's definitely a concern.
In the comparison of functions versus taxonomy,
yes.
I think it's important to point out that if you just take random samples of organisms from across the tree of life, they can have very similar functional contents, regardless of them coming from the same environment or having any type of functional convergence or whatever. And that's because the scales that you're comparing there with sort of taxonomy of genera versus protein functional diversity are very different. So do you correct for taxonomic sampling when you do those comparisons?
So that's certainly a fair point. That you know, all life has sort of a core functionality in common, or larger scales have core functions in common that would make this show up. One of the things that was seen in a lot of the HMP work is that you see this pattern over and over, where within a body site, even though the phyletic distribution can be very different, the functions are quite similar, but then if you compare that functional profile between sites, it can be quite different. So it's not simply the case that all bugs all do all the same stuff, but you do see sort of different levels of resolution.
But I guess some people here, Greg and Rob Knight and others, have shown that you can predict a lot about the functional diversity from the phylogenetic distribution that is present in a sample. So the differences between different sites might be partly due to different taxa present in those sites, and might be partly due to different functions. So you need to sort of find, we as a community need to find ways to sort of figure out how to sort this out.
Absolutely, and there's questions of how well we've actually covered the functional space as well, and versus taxonomy. So definitely a fair point.
Thank you so much.